Hi class, this is the Module 13 lecture. The topics is genetics and inheritance. But before I get started, look, there's Meatball, looking right at you. He's the biggest of my three cats. He's 22 pounds. All right, so we are in chapter 9 of your textbook. And we'll go through the entire chapter. So we'll spend a lot of time talking about the different patterns of inheritance. Okay, so first of all, the genetics began way before the tools that we have available today were available. Nowadays, we can look at a gene sequence or a protein sequence as a way to study inheritance. But back when Gregor Mendel was working with genetics, this was in the 1860s. Those tools weren't available yet. He did his work largely with plants, and he was able to even figure out back then that something was being passed from the parent plant to the progeny plants. So he called, this is heredity, is the transmission of traits from one generation to the next. Mendel called these heritable factors. We now know them as genes. So genetics is the study of heredity. And as I mentioned, Gregor Mendel was doing his work in the 1860s. He had a background in physics, chemistry, and mathematics, and he also had a love of the garden. At about the same time, Darwin was coming up with his theories on evolution. And I think it's actually quite interesting that both of these people really came to the same conclusion, that there are heritable factors that are passed from parent to offspring, but there's different types of control on the expression of those heritable factors or genes. So both of these men were studying different aspects, but their deductions supported each other. So by 1866, Mendel had described correctly that parents pass on heritable factors to their children, and these are known as genes. And furthermore, these genes are retained in a person or in a lineage, generation after generation after generation. So we call a character a heritable factor that varies among individuals. So flower color is an example of a character where the trait would be the actual color, white flowers versus pink flowers versus purple flowers. So the trait is a description of the character. Maybe that's a helpful way to think of it. So what he did is he took pea plants and controlled the mating of them. So he was attempting to cross-fertilize plants, and he did this by preventing self-fertilization. Some plants are capable of self-fertilization as well as using forms of sexual reproduction. So Mendel wanted to look at these heritable factors being passed from parent to offspring. So he did this by preventing fertilization, self-fertilization of the pea plants. And he specifically did this in the male pea plants. I'll show you a picture in just a moment. So this was done prior to the production of pollen, so it can't self-fertilize. Then he took that pollen and painted it onto the carpel, which is the female reproductive organ in the pea plant. And this was the fertilization event. So after pollination, the carpel, these are the different terms for the parts of the plant, well, developed into a pod, and inside the pod are the seeds, otherwise known as peas. And then he planted the seeds and studied those. So that's the basic setup of his experiment. So here is a picture of the garden pea plant. The carpel is this longer structure, and this is the female structure that contains the eggs. And then around it are the stamen, which at the very tip have a structure that contains the pollen. So the way that I remember that the stamen is the male reproductive organ and the carpel is female is, look at the name, stamen. That's how maybe that'll help you. So that's the anatomy of the plant. So what he did is he cut off the carpel, or excuse me, cut off the stamen to prevent self-reproduction, and then transferred pollen from a plant to his original one. So he used a plant that had purple flowers and a plant that had white flowers, transferring the pollen from the white flower onto the carpel of the purple flower. And then they grew up into seeds, and then he planted the seeds. So we use P for parental generation. And then the offspring are called the F1 or F2 or F3, depending on which generation we're talking about. So F is for product of fertilization. So this is the product of fertilization round 1 from these 2 parent pea plants. So that's the technique. So now let's take a look at the different characters that he was able to study. So he looked at flower color. He looked at the position of the flower on the plant. Is it in an axial position, branching off, or is it in a terminal position? He looked at seed color and shape, pod shape and color, and then the length of the stems. And he identified dominant and recessive traits for these characters. And those are as listed. So in order to do this properly, Mendel needed what is called a true breeding variety. And this is simply a result of self-fertilization. So all the offspring look just like the parent because there's no genetic mixing from 2 sources. We're looking at the genetics of one parent being passed to progeny. So true breeding parental plants are the parental generation again. And then we get the genetic cross. So that's the product of fertilization, which is also known as hybridization. And here, the offspring are hybrids when they bring together 2 varieties. The hybrid offspring would be the F1 generation, product of fertilization 1. And then if we take 2 members of the F1 generation and mate them together, it produces the F2 generation. So this leads us first to what is called a monohybrid cross. And this is mono, meaning one or single. This is a cross between 2 individuals that only differ in one character. This is the simplest of the genetic crosses to do. So he did this with a plant with purple flowers and then another plant with white flowers. So those are the parentals, and this is what he found. So here we have the purple flower mating with the white flower. This is the P1 generation. The product is the F1, and all of these flowers, interestingly enough, had purple leaves. But when he took 2 members of the F1 crossed with F1, now he sees a different pattern. The F2 generation had 3 out of 4 plants with the purple petals, but 1 of the 4 plants had white flowers, even though none of the F1 had white. So this is the data that allowed Mendel to see that something is being passed from progeny down into the successive lines of fertilized generations. And we see that with the reappearance of the white flowers in the F2 generation. So this is an example of a Punnett square. And I put this in here so that you would have a blank one to work with, or to see how to fill them in. So the way that we do these is these are the alleles that one parent has, and then we have the alleles of the other parent. And then the cross would be big P and little p, big P and little p, big P and little p, big P and little p in the first box. But then if we take those, big P, little p, and made it with big P, little p, we can do the same thing and fill in the boxes again. So I do encourage you to practice doing Punnett squares, because it is a good way to learn and understand that you are getting these genetic concepts. Your textbook has a few of them in the chapter, and then you can find the answers for those in your textbook as well. So that's nice. You can double check your work. Okay, so filling it in, this is what it would look like. So our F1 generation, a purple plant crossed with a white plant, produced an F1 where all the flowers were purple. And they had one dominant allele represented with capital P and one recessive allele represented with the lowercase p. I put it in italics, the lowercase p, just to make it more obvious that it's a lowercase letter, since they look similar. But all four of the progeny here are the same in the F1. But then we take two F1s and cross them. So big P, little p, big P, little p. And here's our pattern that we get. So we have 4 plants, 3 of them, 3 combinations produce purple, or it's actually 2 different combinations. And then we have a third genotype and phenotype. So these might be new words for us. The genotype is the genetic combination. The phenotype is the physical appearance. It's what you see on the outside. So here in this example, the phenotype would be a white flower, whereas in the other 3 boxes, the phenotype is purple. So what Mendel was able to demonstrate in this simple experiment is the pattern of dominant inheritance. Simply put, one big P is enough. If we have one big P, we get the phenotype associated with that trait. If we have 2 big P's, we still get the same phenotype. It's only the recessive form that has a different phenotype here. So this brings us to the law of segregation, and what this well, let me show you. I'll show you as we go through it. So he was at the time, again, none of this had been worked out. So Gregor Mendel's trying to figure it out. So there was another hypothesis proposed, which was a blending hypothesis. But that's different from dominance. So what Mendel still needed to figure out is what happened to that white flower color. It disappeared in the F1, but then reappeared in the F2. Also, he wanted to explain the ratio. In a monohybrid cross, we see this same ratio every time in the dominant patterns of inheritance, where 1/4 of the F2 have a different phenotype than the other ones. Okay, so he ended up developing 4 hypotheses, and we'll describe them based on the current nomenclature. So alleles are alternate versions of a gene, and they do account for variations in our inheritance. So we inherit 1 allele from our mothers and 1 allele from our father. So those could be the same, they could be different. So an allele is an alternate version of a gene. For each character, the organism can only have at most two alleles, 1 from each parent. And again, they can be the same or they can be different. A homozygous has the same two alleles. So two big P's or two little p's. The heterozygote has a difference, big P and little p, using our example of the pea plants. Third, the law of segregation applies to this pattern of dominance. So if the alleles, if we have different forms, different alleles in the pair, the dominant allele is what wins out in the phenotype. So we've got dominant and recessive alleles. And again, phenotype is the physical expression. So the gene is the nucleic acid. The phenotype is equivalent to the protein. It's the product of the gene. And the genotype is just the individual genetic makeup of that particular trait. And like we saw with the purple plants, we can get the same phenotype with more than one genotype sometimes. Big P, purple plants. Big P, little p, purple plants. Those are two different genotypes for one phenotype. And then finally, the law of segregation. And what this means is that the sperm or the egg has only one allele due to meiosis. And these allele pairs, think back to what we learned in our last module, they segregate during cell division. And that is what the law of segregation is. And then at fertilization, we bring back together the gametes and get an allele pair again. So we're talking some other words we could use for this are diploid and haploid. So Mendel's hypothesis does explain the ratio that he saw in the F2, that 3:1 ratio. So we take our F1 with the heterozygous genotype and cross it with another F1, and then we see that characteristic pattern that we looked at. So our Punnett square is a way that we can show the different possibilities of gene or of allele combinations that could occur when the gametes come together. Let's run through it again. So now we're looking at an alternate explanation of the crosses. So here we have the same setup. We're taking a purple flower and a white flower and mating them to produce the F1 generation. So these would be big P and little p. That's not new. That's what we saw previously. And then we get our 3:1 ratio. So what big P and big P gives us the homozygous dominant. Little p and little p gives us the homozygous recessive. And then big P and little p give us the two heterozygotes. So in looking at the F2 generation in the dominant patterns of inheritance, 3 of the 4 predicted progeny would have purple flowers and only 1 of the 4 has white. In this example, we have genotype 1, genotype 2, genotype 3. So we have 3 genotypes, but 2 phenotypes because of the dominant pattern of inheritance. As long as there is one dominant allele, that is what the phenotype will show. So it held up. He could do this over and over again and get the same ratio every time he did the experiment the same way. So a locus it's derived from the word loci or location. It's the specific position of a gene on its chromosome. And each gene has its own specific locus. And then when we're looking at a pair of homologous chromosomes, having moms and dads one, the homologous chromosomes would be that locus would be the same in a pair of homologous chromosomes because they're the same chromosome. So with our homozygous individuals, they would have the same allele on both homologs, where the heterozygote needs to have both. Okay, so let's look at an example of this. So we've got two homologous chromosomes. We'll just say we've got our blue one and our red one, just to keep it simple. And then we're looking at three different locus or loci. We've got the P locus, the A locus, and the B locus. So we will run through these one by one. All we can get by crossing big P with big P is big P, the homozygous dominant genotype, which will lead to the dominant phenotype. When we cross little a with little a, we've got the homozygous recessive expression. But now with locus B, we've got the heterozygote that has one dominant and one recessive allele. So what's happening at one locus here has no bearing on the other locus. All right, so we've been looking at monohybrid crosses, but there is what's called a dihybrid cross. And this is when the parentals differ in two characters. A dihybrid cross is the same thing as two monohybrids put together, but we can just do it as a dihybrid cross as well. So Mendel did this. Now he was looking at two characters. So we've got round yellow seeds, and then we've got wrinkled green seeds. So we've got color and shape of the peas. Those are the two characters in this dihybrid cross. The F1 generation all look the same. So these are all plants that have yellow round seeds, and we'll see the example on the next slide. But now in the F2, he got a different ratio. 9 out of 16 had round yellow, 3 out of 16 had wrinkled yellow, another 3 out of 16 had round green, and only 1 of the 16 had wrinkled green. 9 to 3 to 3 to 1. This is our ratio for a dihybrid cross in the dominant pattern of inheritance. The F2 will have 9 to 3 to 3 to 1. So what we see, we're looking at a hypothesis on how these different characters segregate. And what we see is this box. It's a lot to look at because we are following two different characters, and then we have the variants of each one. But this is where our 9 out of 16 are yellow and round, so you can count them up. And that's because these are the dominant versions of these two characters. We've got yellow for the dominant color, and then we've got round for the dominant shape. And then you can fill it in as you go through and do these crosses. So here's our ratio that holds true over and over again for dominant inheritance of two characters in a dihybrid cross. What was refuted is the hypothesis of dependent assortment, which didn't give us the right ratio, right? We didn't see this 3 to 1 ratio. So what this showed us is that what's happening on the R locus is independent of what is happening on the Y locus, even though we're looking at aspects of the same seed here. So the hypothesis of independent assortment is what was proven as being supported, because this ratio of 9 to 3 to 3 to 1 kept coming up over and over again in the F2 generation. So similarly to what we went through with the monohybrid cross, Mendel needed to explain this ratio in the F2 of the dihybrid. He's got new combinations here that weren't seen in the parents. either of the parents. Okay. So what Mendel did is he suggested correctly that the inheritance of one character has no effect on the inheritance of another. So the inheritance of seed color has nothing to do with the inheritance of seed shape. Those are independent events that are happening. And in doing so, why don't you try to do it? Try to separate out a dihybrid cross into the individual monohybrid crosses. There would be 2 of them for a dihybrid cross. Okay, so this is the law of independent assortment. Okay, so we can see this in this example with these really cute puppies, or Labradors, I should say. So let's look at our phenotypes and genotypes. So we've got a black coat with normal vision. We have a black coat blind, we have a chocolate coat normal vision, and then we have a chocolate coat blind. So blindness is recessive. So that means we need to have the homozygous recessive pattern for the vision here. And then also the brown color is recessive. So in order to have the brown colored fur, we need to have the homozygous recessive expression of on fur color. So when we take these and mate them, so we're mating the double heterozygotes. So that would be big B, little b, big N, little n, again, crossing itself. Look at the pattern we get. 9 of the dogs have a black coat with normal vision. 3 of them have a black coat, but they are blind. 3 of them have a chocolate coat with normal vision. And 1 of the dogs has a chocolate coat and is blind. And this is showing us the law of independent assortment. In this case, we are looking at fur color and vision. Okay, so a test cross is actually a really useful, simple way of trying to identify heredity or trying to figure out a potential gene. A test cross is always done with the homozygous recessive, always. So it's a way to try to figure out the genotype of an unknown organism by crossing it with the homozygous recessive, because the only way you can get a homozygous, you can get a recessive progeny would be if the unknown also had the recessive allele. Let's take a look at what I mean. So it is a test where we can identify the genotype as either having a recessive allele or not. And this is based on the probability, the numbers that come out of it. So Mendel did this over and over again to verify that he had true breeding plants. That would be really important in his experiment. So if we're trying to figure out the genotype of the black dog, we know that it has at least 1 dominant allele to be black, right? It could be big B, or big B, little b, and still be black. But here's our homozygous recessive. And we're only looking at fur color here. We've gotten the blindness out of it just to make it a little bit simpler. So we've broken up our dihybrid cross into a monohybrid cross. So let's look at the 2 possibilities from this cross. So if we take big B, and cross it with the homozygous recessive, which would be a little b, right? That's all there can be donated is a little b. Our F1 is all black with the heterozygotes. But if we have big B, little b right here, big B, little b, and we cross it with the recessive, now we have the opportunity to see that recessive phenotype, which tells us that the dog, and if this is our outcome, it means the parental was homozygous, was the heterozygote when mixed with the homozygous recessive. So that's the utility of a test cross, and it is a way to help figure out the genotype of an unknown organism. We actually used to do we did a variation of this experiment at the University of Arizona when I taught there. We would do it with blood typing. The students, medical students would blood type themselves. And we actually got told by the attorneys, by the university attorneys, not to do it that way anymore, because one of our students unfortunately found out that his parents were not the parents he knew them to be. Can you imagine? That's a pretty harsh way to find that out. So a trait does not necessarily, let's talk about what dominant means. Dominant does not mean normal. It doesn't mean abnormal. It just means it's more common, because there's more 3-to-1 are going to be dominant in a monohybrid cross. So wild type traits, that's a fancy word for saying that those are the type most often seen in nature. But it doesn't mean it's dominant, because we do have examples of wild type traits that are recessive too. So let's look at some of those examples. So dominant is the presence of freckles. Recessive is no freckles. But look around next time you're out. Most people don't have a face full of freckles, because the wild type is no freckles. That's what's more common. And then we've got the example of albinism. So we've got a man with normal pigmentation versus a man that has albinism. And this is less common. Albinism is less common than normal pigmentation. So we don't want to get into the mindset of thinking that a dominant allele is somehow better than a recessive allele. It isn't. It just means it's more frequent. Okay. We do have a number of genetic disorders, and some of those are caused by one single gene. Many of them are caused by a combination of genes. And that's where it gets really hard to study. The single gene genetic disorders are a little bit easier, because you don't have to take into account all these other factors. So albinism is an example of that. But also so is cystic fibrosis. These are both examples of inheritance of recessive traits. People that have cystic fibrosis, this is a condition that affects the lungs. People with CF produce a lot of mucus in their lungs, but also their cilia don't work right. Remember that the cilia help to beat and move material out of the lower respiratory system that shouldn't be in there. So people with cystic fibrosis have very thick mucus and an increased production of it, and their cilia don't work to help expel it out. So it's a condition that most people, you don't see many cystic fibrosis patients in their 40s or 50s. Most CF patients die before then of a type of pneumonia or oxygen deprivation. Okay, what we haven't talked about yet are carriers. And a carrier is somebody who is a heterozygote. So that means they have the recessive gene, but they're not sick themselves. They have the genotype of the recessive allele, but the dominant allele wins out again. So let's look at this example using albinism. So our parental generation, we have heterozygotes. So big A, little a, big A, little a, and we get our 3-to-1 ratio. So 3 of the 4 offspring, or 3 of the 4 combinations would show the dominant pigmentation, which is normal, and only the homozygous recessive shows the phenotype of albinism that has that lack of pigmentation. So same ratio that we saw before with a monohybrid cross, 3-to-1 or 75% to 25%. One thing I'd like to say is what this does not mean is that if a person has 4 babies, 3 of them are going to be normal and 1 of them is going to have albinism. We can't say that. What it means is that the chance of the progeny having normal pigmentation is 75% and 25% for abnormal. So you don't want to divide it out by children, if that makes any sense. We'll look at an example of this later on too. So here's a list of some of the common autosomal, so that means non-sex chromosome. That's what an autosome is. So these are some of the common genetic autosomal disorders that are in humans. So here's cystic fibrosis. Sickle cell disease is another one, and that results in abnormally shaped red blood cells and can impact oxygen distribution. Huntington's disease is a dominant disorder, genetic disorder, that impacts the nervous system. And then even some causes of high cholesterol have a genetic link. There's actually several different causes of high cholesterol. And that's why one medicine may work in a person and another doesn't. It may take trying different medicines, because if we're talking about the genetic version of the disease, well, that's a different mechanism than, say, obesity-related high blood pressure or in high cholesterol, or diet-related. Okay, so we've been talking about the dominant pattern of inheritance. Now we're going to switch off and talk about the other two patterns of inheritance. So complete dominance, that's what we've been talking about, just to make sure we understand the terms. Another pattern of inheritance is incomplete dominance. And this is where the hybrid is somewhere in between the two parentals, kind of like a blending. And we can see this with snapdragons. My snapdragons just died, otherwise I would show them to you. I grew them for this purpose. So we're looking at a red flowered snapdragon, dominant, and a white flowered snapdragon, recessive. Now look at this. We have a new phenotype in the F1. We have pink. They are all heterozygotes, but we have this phenotype that is in between both of the parents. That's why I think of it like a blending. And then if we go through and mate two members of the F1 generation together, we end up with this genotype and phenotype at the bottom. So 3 of the 4 of them have the dominant allele, but only the homozygous dominant is red. The heterozygote is pink. The homozygous recessive is white. So now we have 1, 2, 3 genotypes. We also have 3 phenotypes. That is a difference from the complete pattern of inheritance. There we only have 2 phenotypes with 3 genotypes. Here it's 3 genotypes, 3 phenotypes, because of this intermediate pink flower. Okay, so an example of incomplete dominance in humans is with this genetically linked elevated cholesterol, which is hypercholesterolemia. And these can be quite dangerous, or this condition can be associated with quite dangerous levels of cholesterol in the blood. And interestingly, heterozygotes have intermediate cholesterol. So that tells us that it is an incomplete dominant pattern here, because if it was dominant, the heterozygote would be the same as the homozygous dominant, but it's not. Think of the white flowers mating with the red flowers to give us pink. So here's our pink. The heterozygotes have an intermediately high cholesterol, where recessive people wouldn't have it, and homozygous dominant people have even higher. So that is the incomplete pattern of dominance. Now, we've been talking about really just two alleles so far, because that's all an organism can have. But in a population, there can be many other variations of a gene, many other alleles. So this is bringing us to the co-dominant pattern of inheritance. Right, only a person can only have 2 possibilities, but the population can have a lot more. We'll look at blood typing as an example here. So the human ABO blood group is an example. There's actually two patterns of inheritance here. There's dominance and then there's codominance, but it is considered to be a codominant expression. So we have 4 different blood types. There's blood type group A, type B, type AB, and type O. And we've got these genes, these alleles, the A and the B, and then the recessive form. We don't call it O, it's actually it's represented with a little I when we're looking at the genes. So in the AB group, this is the classic example of codominance because these red blood cells have the A sugar and the B sugar on their cell surface. That both alleles are expressed at the same time. Codominance. So let's get to a picture. It's much easier to see here. So type A, the genotype would be I to the big A, I to the big A, or here's that other one, that recessive form. And we represent it with the little I. So having one big A is all we need to get carbohydrate A on the cell surface of the red blood cell. B is the same thing, but with the B allele. And now these cells have the B carbohydrate. AB, there's only one genotype that specifies the phenotype for type AB, and that is the AB. They have both carbohydrates. And then type O, the genotype is homozygous recessive, represented with 2 little i's. There's no sugar on their red blood cells. Now, the thing about, we haven't gotten to immunology yet. So let me just try to explain this really simply so that we can understand what you need to know for the purpose of genetics here. A person that has type A, they don't react to blood type A if they're given it, but they will react to blood type B and type AB. So let's look here. So when blood type A is mixed with type O, nothing happens. This is a negative reaction. When it's mixed with blood type A, nothing happens because this person has type A. They don't have pathogenic immune responses to type A. But look, in type B and type AB, it's different. This is a positive test. It's called agglutination. It means that the pathogenic anti-B antibodies that are in this person's blood react with B. They also react with the B part of AB. Okay, so this is why it's a little confusing because when we think of antibodies, most people think of them as being raised in response to an infection. So there's something good that protect us. That's actually the example of what's called a pathogenic antibody. But we have other antibodies in our body that are healthy for us. So a person that has blood type A, they actually have antibodies to A as well. They have anti-A antibodies, but they're not pathogenic antibodies. They are called natural antibodies. And this is how our immune system learns not to fight its own blood type because it's learned not to respond to it. It has not learned to respond to B, though, in the A person because this is a different blood type. So there's no way to tolerize the immune system to not respond to B. So hopefully that makes a little sense to you. So AB, look, there's no clumping reaction with anything. AB, universal recipient. They've learned how to deal with carbohydrate A and B so they can get any blood type and not react. And then blood type O, well, there's nothing on the cell surface here. So the O is the universal donor because there's nothing on the cell surface to cause a transfusion reaction there when with type O. Okay. But they still have those anti-A and anti-B antibodies in them because they haven't learned, their immune system didn't learn, hey, don't respond to this because they haven't seen it. So that is codominance. So we've talked about complete dominance. We talked about incomplete dominance and codominance. In reality, though, many of our characters are influenced by a combination of genes and the environment. So skin color is a great example of that, right? I've got light color skin, but if I go out in the sun, it will change colors upon exposure to the sun. Heart disease and cancer are also good examples of complex diseases that have both genetic and environmental causes. A lot of these type of studies have been done by looking at identical twins, because an identical twin has the same DNA. They have the exact same genes. And if it were all due to genetics, then every member of a twin pair would have the same phenotype, but they don't. They don't at all. I can use the example of studies with autoimmune diseases where they've looked at twins where one person has lupus and one person doesn't, even though they have the same genetics. That speaks to the influence of the environment. And that would include things like infectious agents, as well as non-living factors or chemicals and things like that. Okay. So the chromosomal theory of inheritance does describe that genes are at set locations on chromosomes. So every time we look at gene A, it's going to be on the same place on the same chromosome. The chromosomes undergo segregation and independent assortment during meiosis. So if the mom's chromosome 1 goes in one cell, it has no impact whatsoever on what happens with chromosome 2. That's just how they line up on that metaphase plate. His laws are also, they also do explain, they hold true in mitosis as well as meiosis. So a way to apply this is that our allele pairs are going to separate, right, during the process of meiosis. That's the whole purpose of meiosis. It has that extra round of cell division without another replication process, so that it goes from diploid to haploid, where there is a single copy of each chromosome in the gametes. And then the gametes mate and bring back 2 copies. So the law of segregation really is most noticed, of course, during anaphase 1. Anaphase is when the 2 cells start to separate. And this would be the homologous chromosomes if we're talking about anaphase 1. It doesn't even make sense to talk about the sister chromatids here, because a sister chromatid is a duplicated copy of a chromosome. So it only makes sense that the law of segregation is describing the separation of the homologous chromosomes to create the gametes. And then we've got the law of independent assortment. So we've got the law of segregation, which has to do with the formation of the gametes. And then we've got the law of independent assortment, which simply states that what happens with one allele has no bearing on another allele or location. It depends instead on how everything lines up during metaphase. Just like the law of segregation is dependent on anaphase 1, well, that's based on the metaphase alignment. All right, so with our dihybrid cross, another group of scientists, you might recognize this name, Punnett, from the Punnett square. Well, they looked at what are called linked genes. And what they saw is that they didn't get this normal ratio that Mendel got in his dihybrid cross. So they began to look at, well, why is this? And what linked genes are, these are genes that are inherited together. So the closer together they are, the higher the chances that they come as a set, because otherwise you have to cut right in the middle of them. There would have to be a cleavage right in the middle of them, or crossing over to occur right in the middle. And statistically, the probability of that happening is low for linked genes. So they come together. So let's look. Okay, so just to go back to our normal experiment, we've got our purple flower, and now we're looking at purple flower and pollen. So flower color and the shape or length of the pollen. So in this cross, the prediction was 9 to 3 to 3 to 1. And these would be the numbers based on that, but this is not what was seen. Especially look at this red round that should be the lowest. It's higher. So something else is happening here. And so here's the explanation. So we've got two locuses on the same chromosome. So we've got the P locus and the L locus. P is for color, L is for length of the pollen. So crossing the parentals would be the homozygous dominant crossed with the homozygous recessive. And then we end up with our gametes as follows. And then we mate the gametes. So we have the combination of big P, big L with little p, little l crossed with the same possibility for in the eggs. And now we get this 3 have a purple long, 1 has a red round, but we don't have any accounting for the other 2 phenotypes. So this is the data that showed the presence of linked genes. And this doesn't occur with all genes. I mean, some are linked and some aren't, but the closer together they are, the higher the chances that they come as a set. And this is due to crossing over. So if you remember, crossing over is something that's happening on homologous chromosomes. Again, a sister chromatid is just a duplicated chromosome. Even if there's crossing over, it makes no sense because it would be switching the same genetic material since it's a copy. So we're talking about homologous chromosomes, which means we're talking about meiosis 1, because the job of meiosis 1 is to separate homologous chromosomes. Meiosis 2 separates the sister chromatids. So linked genes can be separated by crossing over, and then we get something new. We get these recombinant gametes, and we can figure out the recombination frequency. So let's take a look. Here we go. So here's our homolog. We've got the homozygous dominant on the red chromosome crossed with the homozygous recessive on the blue chromosome. So we've got 2 different locuses, and they're actually pretty far apart in this example. But look here, crossing over happens, and the crossing over is occurring in between the P and L locuses. So we end up, yes, we do get some gametes that are just like the parents. So we call these the parental gametes, but now we've got these brand new combinations that did not exist in either parent. These are the recombinant gametes. So we've got little p with big L and then big P with little l. There's no combination like either of these in the parentals. So we've got 4 possibilities. 2 of them are parentals and 2 of them are recombinant for the gametes. And crossing over is what produces those recombinations. All right, let's spend a little bit of time talking about sex-linked genes. So these are genes that are on one of the sex chromosomes. So for humans and other mammals, XY are anatomically male, XX are anatomically female. There are cases of XXY, that can happen. There are cases of XXX, that can happen. But typically, this is what we're looking at in mammals. So we have our 1 pair of sex chromosomes. And if you're a female, they'll match. But if you're a male, they don't. It's a completely different chromosome, X and Y. And then there are the 22 pairs of somatic chromosomes, non-sex chromosomes. So that gets us to a total of 46 chromosomes. Okay, so in mammals, including in humans, the Y chromosome has a gene called SYR, and it is needed for the development of testes. So without the expression of the SYR, the gene, the developmental pathway goes towards the development of ovaries, which is what it's programmed to do unless this gene is expressed. And then it'll be associated with the development of testes. Okay, so let's run through this figure. This is figure 9.20. Now, some versions of the textbook have a mistake in this figure. So this figure here is corrected. I corrected it, which means that the figure you see in your textbook might be wrong. It has the wrong combination in the gametes, but this is correct. This is the corrected figure. And I did send a notification to the publisher 2 years ago to make that correction. So for a male, sex chromosomes X and Y, and then we have our 22 autosomes, 22 non-sex, and so 22 times 22 is 44. But for the female, it would be XX. So for the male, we have 2 options for the gametes: 22 chromosomes with an X, 22 chromosomes with a Y. Where for the female, she's only got an X, so it's 22 plus X. So if we mate the X from the male with the X from the female, we get a female offspring. And now we're back to being diploid with a copy from each parent, 2 copies of each chromosome. The way we get a male is from the Y chromosome that the male has mixing with the X of the female to create a male baby. So again, please note that some editions of the textbook, I know for a fact it's the one that has the snow owl on the front of it. So I think that's the 8th edition. This figure is incorrect in your textbook. It's got the wrong number in one of these circles here. And it completely confused me when I looked at it. So I'm sure it would do the same to others as well. So sex-linked genes can be on either the X or the Y. They're linked because they're on one of the sex chromosomes. And it doesn't have to have, these genes don't have to have anything to do with sex. Some do, but most of them don't. In fact, the X chromosome has lots of genes on it that have nothing at all to do with sex. So we'll look at the example of eye color in fruit flies. And this is an X-linked recessive trait in fruit flies to get white eye color instead of red. So we've got red eyes and white eyes we're looking at. So this is recessive, this is dominant. So let's run through a couple combinations here so we can see the impact of sex-linked genes. So we have a female, she would be XX because she's a female. And in this example, she is homozygous dominant, being crossed with a recessive male. Now note the color difference right off the bat. The female has red eyes, the male has white. The male has the recessive phenotype, but he only has one recessive allele. That's all he needs because there's not another X there, it's Y. So we tend to see recessive X-linked diseases more so in males than we do in females because the female still needs to have both recessive forms of the allele where the male doesn't, he just needs one. So if we do our cross, we end up with all red-eyed because of the dominance coming from the female. Now we're going to cross a heterozygous female, so she's got both alleles, with a homozygous dominant male. And now we see the white eye again, because now we can get either a X big R, or we can get this combination, X little r coming from the female with the Y of the male. And this would be a recessive genotype and phenotype. So yes, most sex-linked disorders are due to recessive alleles, and they are seen much more commonly in males because it's easier to have the recessive genotype and phenotype when you only need one form of the allele. So a male that inherits a X-linked recessive allele gets it from mother. the father's donating Y. So they're passed through the mothers to the sons, where a female has to have both recessive alleles, as we were discussing. All right, that actually wraps us up on Module 13. These are your study slides. So please take a look when you're putting this information together. These are slides I find are really important in going over some of the main concepts. So I'm also hoping to remind you of the process of meiosis by showing you this slide, and haploid, diploid, so on. And then you've got your flow chart here to fill in. The answers for this are in your textbook. All right, class, I hope that was helpful. I hope everybody's doing well and that you're safe, your families are healthy. Have a great day or evening, wherever it is when you find this. Take care. Bye-bye.